Muy buenas tardes. A nombre del Colegio Nacional y en particular a nombre del doctor José Sarukhán y del mío propio, les queremos dar la bienvenida a una sesión adicional de los viernes de la evolución. En esta ocasión se conjuntaron los astros y tenemos la fortuna de que el doctor Bevilacqua, que es un profesor de Penn State University del Departamento de Biochemistry and Molecular Biology, haya venido a México a visitar una pariente muy cercana y cuando me dijo que iba a estar aquí rápidamente se nos ocurrió invitarlo para dar una charla sobre el campo en el que trabaja, que es un campo con profundas implicaciones evolutivas. Él muy generosamente aceptó. Es día de San Valentín, de manera que es una buena forma de celebrar el Día de la Amistad, del Amor y la Amistad. Y el doctor Bevilacqua lleva muchos años trabajando en estructura y propiedades del RNA. Como todos ustedes saben, se había teorizado, sobre todo muy fuertemente desde 1968 con los artículos de Leslie Orgel y Francis Crick, que el RNA podía tener propiedades catalíticas. Esto fue recibido con un enorme escepticismo por buena parte de la comunidad científica, entre otras razones porque todos habíamos sido educados y seguimos educando con la idea de que quien ejerce la catálisis en las células son las proteínas y no el RNA. En 1986 se descubre que el RNA efectivamente tiene propiedades catalíticas y esto vino a modificar enormemente nuestras ideas sobre el origen de la vida. Y al mismo tiempo que esto fue un descubrimiento muy bienvenido para todos nosotros, los que trabajamos en origen de la vida, y muy sorprendente para quienes hacían biología molecular, se desarrolló una corriente extremadamente reduccionista que aseguraba que en el momento en que había aparecido una molécula de RNA en la Tierra primitiva, había aparecido la vida. Pero se dan ustedes cuenta que esto es equivalente a esa visión simplista de que el DNA es la molécula que guarda el secreto de la vida. Si algo tienen las células es que son un conjunto de moléculas, subsistemas y sistemas que interaccionan entre sí de una manera muy rica. La pregunta es, ¿y cómo surgieron entonces en la Tierra primitiva este tipo de sistemas polimoleculares? El doctor Bevilacqua venturosamente pertenece a la corriente que sostiene que, contra lo que se afirma en el reduccionismo molecular, la vida no depende de una sola molécula, que a pesar de las propiedades tan asombrosas del RNA, estamos viendo un componente esencial, pero no único, de la esencia de la vida y de que en la Tierra primitiva debe haber habido interacciones entre muchos tipos de compuestos adicionales. De ahí el título de su charla, RNA and Protocells in the Emergence of Life, el RNA y las protocélulas en el surgimiento de la vida. Antes de darle la palabra al doctor Bevilacqua, permítanme nada más subrayar una palabra que a veces nos pasa desapercibida, emergence, el surgimiento, no con la idea del origen repentino de la vida, sino como parte de un proceso. Y el doctor Bevilacqua en ese sentido pertenece a ese grupo de personas con una formación en biología molecular, en genómica, en estructuras subcelulares, que están haciendo un papel esencial, fundamental, en llevar las categorías darwinistas al mundo subcelular y al mundo químico. Estoy seguro que todos vamos a disfrutar enormemente de la charla de esta tarde y desde luego que, dear Philip, you are most welcome to the Colegio Nacional. We feel honored and we feel very grateful for you being here.
Muchas gracias, Antonio. It's a great pleasure to be here, and thank you for welcoming me. I'm looking forward to speaking.
Lovely. Please.
Thank you.
Chloe, you can translate to your father afterwards.
I could follow many of the things that you were
lovely.
saying. Not
Good.
all, but yes.
Thank you.
No, it's a great pleasure to be here and to be in Mexico City. I've been here for a few days, and my daughter's here, and she is in Mexico City, and so it's a great honor to have her here as well. She's on a Fulbright scholarship teaching English. So, and we spent the day today, much of the day today at the zoo, and it was sort of unexpected. We were wandering through the park and said, let's go to the zoo, and walking around and seeing the great diversity of life and the monkeys and all these things. And it was a great sort of background to think about evolution and life, and just a wonderful day, beautiful day to spend. So thank you again very much for having me. And what I'd like to do is to talk to you about our research in origins of life, emergence of life, and the importance of RNA and protocells, the earliest cells that could contain RNA. And I broke the talk up into 3 parts. I'm going to talk first about protocells, and then a little bit about RNA, and then combining them and show you some data and also some concepts that we have on how they might go together. So I'll go ahead here. I think, there we go. So I want to begin by mentioning and acknowledging my close collaborator and friend, Christine Keating, who really pioneers the protocell ends of things. She's a professor at Penn State and my collaborator for over a decade now. And I'd like to begin just by talking about exoplanets. So these are places in the universe where the conditions are such that there could be life. And there are so many that we know about, and more that are being discovered. And some, whoops, sorry, get this right here. There we go. And let's see. Here we go. And they're listed over here, okay? And one of the things that we wonder about is whether there could be life out and elsewhere in the universe. And I'm curious, just for those in the audience, if you want to do this by raising your hand. So how many of you think that it is very unlikely that there is life elsewhere in the universe? So two people. How many of you think it's somewhat likely that there's life elsewhere in the universe? And then how many think it's very likely that there's life? Okay, so most, but not all. So why do we think it's likely? Well, we have to think about the plausibility of life, how it starts, and how it's captured in protocells. So we'll stick with it this way. So we only know that life exists on one planet, okay, our planet, Earth. And so we can think about how we might move from matter to life. And one of the things that we consider is Darwin talked about warm little ponds where the conditions were just right for life to begin. So maybe neutral pH, low salt, and wet, dry cycling. So this is sort of throughout the day when it's warm and the ponds or puddles dry down and the reagents concentrate so that reactions are more likely when they're concentrated. And then it rains again to dilute this system and to allow cycling to happen. And the building blocks for life are found, as I'll show you on the next slides, in a number of different ways, leading to precursors for proteins as amino acids, nucleosides for RNA, and lipid precursors. And the conditions on early Earth provided a number of different locations where life could perhaps start. And they include little areas, like I mentioned, ponds or puddles, deep sea vents that could provide energy for chemical reactions from heat, as well as radioactive sources that can provide heat, and water flow and pores. And so if protocells form, why do we need them? And what are the benefits of having these compartments? If reactions are very dilute, then molecules tend not to collide, they tend not to react. But if we can get them into a tiny little compartment, even liquids inside of liquids for liquid-liquid phase separation, then what happens is that even though a rate constant for two molecules coming together is the same, when the concentrations increase, the reactions can be much faster. And things can happen such as monomers joining together here to make a trimer. And so in these compartments, we get a type of primitive catalysis just by concentrating the reactants. And together with Chris Keating and my lab, about 10 years ago, we showed that RNA could catalyze reactions through simply compartmentalizing the reagents under otherwise very dilute conditions where there's no reactions, shown here, there's no product. We could push them together, and you can see with microscopy, the RNA, the two pieces going in these little cells, and then we can see the product forming. The other thing that compartments can do is protect the molecules from adverse conditions. So out here, they might be subject to UV irradiation, but when they're inside, the outer ones can act as a sort of sunscreen to protect the inner ones. And of course, compartments define individuals and allow evolution to occur. And so if there are many different, say, RNA molecules, as they begin to pinch off and go into individual protocells, then these can start to divide, then the ones that are more thick, shown by the purple colors, can start to populate. So it's useful to think about how modern cells compartmentalize their internal chemistries today. And there's really two ways that we know of. One is by lipid membranes, and that's shown here. And there are many different examples of organelles, the ER and the ribosomes, the nuclear envelope, and they consist of lipid bilayers. But also there are more simple compartments that are formed that don't have membranes on the outside. And we call these liquid-liquid phase separation. So they're liquids inside of liquids, almost like oil drop type of situations. And we know about these in the cell, and there's a number of these, such as P-bodies, stress granules, and the nucleolus. And, oops, wrong direction. And so we can look at some of the characteristics of these. Lipid membranes are formed from fatty acids or phospholipids that can have two fatty chains, and these can come together to form a true lipid membrane containing compartment with a lumen inside and an aqueous solution on the outside. And we can see here that the phospholipids and the fatty acids come together. And they have a number of positive as well as negative characteristics. They're like cell membranes. They can grow and divide. They're semi-permeable. They're very good at retaining their contents. But they have poor encapsulation yields. It's hard to get RNA inside of them. And RNA loves magnesium, and yet these are very sensitive to magnesium and can fall apart. Nonetheless, we've seen something as simple as a 10-carbon fatty acid can begin to form little vesicles, as shown here, and even extracts from meteorites, some of the lipids that are present begin to form little vesicles. But I'll focus mostly on the coacervates, the liquid-liquid phase separation. Also, we call these droplets, so just liquids inside of liquids. I might refer to this as a droplet. And these can form two different ways up in this corner, in a non-associative way and an associative way. If I go back, one slide here to this demonstration of catalysis, Up here, this was in the non-associative way. So these were an aqueous two-phase system. There's no sort of charge interactions. But most of what I'll show you now are through charge-charge interactions. So small cations or polycations and polyanions, opposites attract, so the blue and the red are going to come together and make this coacervate phase. And what drives this, even though these two are coming together and reducing the entropy there, is they're releasing counterions, and that helps to drive this coacervate phase. And you can see the droplets here under the microscope. There are many of them, and they're different sizes. They tend to be on the micron scale, and they have positive and negatives as well. In the positives, they're like these intracellular organelles. Again, they concentrate biomolecules. They provide new microenvironments, but they're hard to maintain individuals. And so the RNAs tend to come in and out of these more readily. We can describe these with phase diagrams, and with different polyions and temperatures, we can tell whether it's going to separate into two phases or if it's just going to be one phase, and we can tell what the contents of the two phases are going to be. And these can be made out of a number of different components. And you can see that they're, as I mentioned, positively charged or cationic polymers, and then negatively charged ones that are going to come together. And the compartments themselves can even be made out of RNA. So these coacervates have a long history in the theories for prebiotic chemistry. Even going back to 100 years ago, De Jong coined the term coacervate from co, together, and aggregation, forming a heap, and was able to see these under the microscope from ground mass of protoplasm, exhibiting properties in common with coacervates. And Professor Oparin, in Origins of Life about 100 years ago, postulated that coacervates could serve a role in the origins of life. And it predates, amazingly, understanding of the discovery of DNA, of lipids, of proteins, DNA structure, and so on. So the final slide, I think, of this section is then that condensates formed by this liquid-liquid phase separation are common in modern cells. And so this is what we know. We can see here modern cells, we can see liquid phase separation, and we can see that we can mimic these with fairly simple polymers that were present in meteorites and on early Earth, as this example here, dependent on these certain conditions. So let me now change, okay, so we have protocells. Now we talk about RNA, and then we're going to put the two together. So shown here is tRNA, transfer RNA, and it forms the well-known cloverleaf structure with the 4 different stems. And these are just some kind of a collection of different tRNAs, quite similar in structures, but subtly different. So one of the things that's happened over the last 20 years or decade in particular is chemists, organic chemists, have played a really important role in showing that it's plausible to go from matter in very simple molecules, simple diatomic, or these consisting of only 2 or even 1 element, 2 different elements, forming much more complex molecules, and then under these conditions. And the famous Miller-Urey experiment in either neutral atmospheres or reducing atmospheres, where these compounds are taken and put together in a spark discharge. This is sort of the equivalent of kind of the mad scientist experiment, where you're really zapping, simulating lightning in the atmosphere, zapping these together and letting this go for a period of time, days or weeks, and then collecting and seeing what comes out. And amazingly, what comes out are these intermediate, growing in complexity molecules, and you can see these unsaturated bonds, these double bonds and triple bonds that we can add to and make more complex molecules that ultimately lead to the building blocks of life. Amino acids that are going to make up our proteins, and then the 2 types of bases that are going to make up RNA, the smaller pyrimidines, C and U, and the bigger purines, G and A. So this is one scenario where the building blocks of life could come on Earth, but maybe they were transported from elsewhere in our solar system or in the universe. And so there's evidence for this as well. Most famously, the Murchison meteorite, which was discovered in Australia in 1969. It's a pretty big rock. It was greater than 100 kilograms, and it's very carbon rich. Analysis of this meteorite also revealed these amino acids, certain amino acids, not all 20, 9 in particular, that are shown here, as well as the 4 nucleobases that make up RNA and also make up DNA, and other ones, related ones as well. And very recently, just in the last 2 weeks, was published a paper analyzing Bennu, the asteroid that was collected by OSIRIS-REx, the spaceship from NASA that went all the way, flew to this asteroid. This is the regolith that was dispersed by OSIRIS-REx. This is a picture of Bennu from 85 miles away. It's pretty big. It's about 500 meters wide, but only it's some hundreds of milligrams, I believe, of material was brought back, and it was parachuted down into the Utah desert a few months ago and immediately put into a nitrogen atmosphere, so it couldn't react with oxygen in our atmosphere. And this was collected at the height of the pandemic, and so a lot of us were isolated away from work, and you had a chance actually to watch this happen and then come back. It landed in the Utah desert very recently, a few months ago, and the analysis revealed, amazingly, again, the amino acids that make up proteins as well as the nucleobases that make up RNA. So RNA can come from, and amino acids can come from simple, very simple molecules that are comprised of only 2 atoms, making more complex intermediates, making these stable building blocks. So let's just talk for a minute about RNA and why it's such an amazing molecule. This is the central dogma of molecular biology, something that we learn probably in grade school biology, and it shows the flow of information, DNA ultimately ending up in proteins, going kind of through the RNA middleman. Of course, we have replication of DNA, but we realize that it's much more complex. Not that the central dogma is wrong, but that it can be elaborated with all these different steps. And these are all RNA-centric steps. So RNA can regulate transcription of going from DNA to RNA, translation of going from RNA to protein. RNA can regulate degradation. RNA can also go backwards to DNA in reverse transcription. This happens in retroviruses like HIV. And then RNA can also replicate itself without going through DNA, like in SARS-CoV-2. So it's quite complex, and that leads us in my lab to kind of view the world this way, which is a very RNA-centric view of that is now kind of the molecule of central importance. As a chemist, and anyone who appreciates art and an artist, we can admire the structures of these molecules. And so over here is DNA, very important molecule, but DNA tends to always form this B-form double-stranded helix. RNA is quite different, and it can fold into these complex protein-like structures, and we can see the similarity that RNA in many ways is more protein-like in its structure than DNA. And it's not because RNA has a 2' hydroxyl, and it's not because RNA and DNA differ by a methyl group. It's because RNA is single-stranded. And as soon as we pair up the 2 strands together, they're tied, and they can't really do much else other than this helix. But RNA can have a much more elaborate structures, and we can see little helical regions in RNA too. But when we look at this, we see that, again, that the RNA in many cases is more protein-like. So we'll talk just for a minute back to tRNA, and I showed you the cloverleaf structure when I began the section on RNA. That's a two-dimensional structure. That actually folds up into a three-dimensional structure that is more like L-shaped here, and that function of RNA follows the form and the structure. And so down here at the bottom is the anticodon, of course, which reads out the codons on the mRNA, and the acceptor stem that will take the amino acid and allow it to be incorporated into the growing chain that is to become the protein. And here's a three-dimensional structure. And I just want to share with you my personal view of RNA and why I think RNA can do so many amazing things. It's because it does what DNA does and then so much more. So just to give you an example, here is an example of what is not traditional or canonical base pairing. We'll call it non-Watson-Crick-Franklin base pairing. So here's your standard base pair. If you look at these two bases, it's just a regular CG, but then we see a third base come in to form a triplet. And I've highlighted it here in ball and stick form and shown here as well. But that's just one example. Here's the same base triple with the GC and then the third base coming in. But there are many non-Watson-Crick-Franklin examples. Here's, of course, a G pairing with a U. Here's a G pairing with a C, but it's no ordinary GC. It's not like that one, which has 3 hydrogen bonds. This is reverse and has 2. And then sometimes we can even get cases where we don't always have a big base with a small base, a G pyrimidine and a purine, but we can have 2 big bases come together. We can see 2 rings and 2 rings. And so we've come in the lab to sort of think of this a little bit more like a Picasso, and who famously said that everything you can imagine is real. And we've taken this to the limit, and even to draw structures that violate, just like Picasso would, the traditional rules. So we don't necessarily set up hydrogen bond donors and acceptors, but even put a donor opposite a donor and look for those, and they can be populated by alternative protonation states, charged states, and tautomeric states. So I like to often look at a scorecard of DNA, proteins, and RNA and ask how they stack up next to each other. First, which can have genetic material? Well, we know DNA can replicate, but RNA can also replicate. I showed you that. And RNA can even go backwards to DNA. There is no reverse translation for proteins. If we ask what can be an enzyme, well, of course, most modern enzymes are indeed proteins, but RNA is also an enzyme, and that was the Nobel Prize in Chemistry in 1989 to Czech and Altman. And if we look at which can bind metabolites, of course, proteins can, and that can lead to feedback and regulation. But RNA can too. This is shown there's a metabolite right there that the RNA is binding. And so if we stack all these up, the only one with a column with all positive charges is RNA. And so that leads us to the notion of an RNA world where we have one molecule that can solve the chicken and egg problem of which came first, the enzymes, which were thought to be proteins, or the genes in DNA. And, well, how about RNA? Because RNA can be an enzyme and a gene. And so I always like to look at Venn diagrams whenever I can. They're useful ways to think. And this, for me, I think we published this in a review a number of years ago. And inside of this lilac color is what's possible, what obeys the laws of the universe, of physics and chemistry. And life must be contained in there. It obeys those laws. And then we have what is, all of us in the room, biology as we know it, molecular biology. And then we have what we really don't know, what was in the RNA world before life really started. But we know it must be inside of what's possible. And then we ask how much overlap. We want to know how much overlap is there between the RNA world and what is. Is there any overlap? And so I want to ask this in terms of a function of the Great Oxidation Event. And so this was an event that happened about 2 billion years ago when photosynthesis began, primarily with cyanobacteria, and oxygen came on the scene. And we know this caused the greatest extinction event on Earth's history. But it also probably caused extinction not just of life, but also of RNA and RNA processes, because RNA is quite sensitive to oxygen. And so we're proposing that perhaps if we look at the overlap here, how much overlap there is between the world that was comprised of RNA and our RNA protein, RNP world, that overlap was high until about the Great Oxidation Event, and then it dropped off to where there's very little, but still some overlap. I'll show on the next slide. But perhaps anaerobes that are not exposed to oxygen, they continue to have more overlap. Something that can be looked at both computationally and experimentally. So let's look at clues for this model. We'll go to RNase P, which was one of the two enzymes that led to the Nobel Prize in chemistry for discovery of catalytic RNA. RNase P trims the 5' end of the tRNAs that I showed you. And so in the tree of life, we can look at structures, and I'm just going to, this is in this review article from Lay. If we look at bacteria over here, and we can see the RNA in gray, we can see there is a protein, but the protein's not very big, and this still acts as a ribozyme or an RNA enzyme. As we move towards more sort of recently evolved species, even archaea up here, we can see it's coated with proteins, the colored species, as if they're protecting the, in some cases, the RNA from oxygen. And then if we get over here into human, we can see there's RNA there. You barely can see it. It's behind the protein, and also with this related enzyme RNase MRP. In fact, you can go so far that if you look at the human enzyme shown here, the tRNA, which is the substrate, remember I said it works on tRNA, is in the warm colors, and the RNA is in the, in the green color. We can see all the proteins that are here. And then something amazing happens. The RNA goes extinct in the most recent species. So if we look, and these are cases where there is a lot of oxygen. So if we look in plants, Arabidopsis, which has reactive oxygen species from photosynthesis, there is no RNA at all. There's only the substrate RNA. The enzyme has become a protein. And same thing in human mitochondria, where respiration occurs and there is extensive reactive oxygen species. The RNA is gone. And you have this thing called ProRP, which stands for protein only, RNase P. So this one's still a ribozyme and RNA enzyme, but these are now protein enzymes. So we wonder how many other ribozymes have also gone extinct. So I told you about protocells and about RNA. So now we'll put the RNA inside the protocell, or at least we'll try to, and we'll see what behavior it has. This is work that's done in the lab. This will be work that's done in my lab in collaboration with the Keating lab. So back to the primordial soup from the Early Earth Company. And here we see what I already showed you with the different ways of building these unsaturated compounds that can react to make the precursors of proteins and of RNA. And here we are again with the associative coacervates from charge-charge interactions. And a former postdoc in my lab, Raghav Padayal, he, in collaboration with the Keating lab, formed these associative coacervates between the positively charged and the negatively charged species. And here you can see they're on the micron scale, these protocells. And what Raghav did was to take a ribozyme that was 2 pieces, and on one piece he put a green fluorophore. We can see the green species here. and then looked at how much more concentrated the RNA became inside of these droplets, and it increased 5,000 times. And so he was able to show that the rate of the reaction increased proportionately compared to in bulk. You know, one of the things about studying early Earth chemistry that I find challenging but rewarding is we don't know exactly what those coacervates were made out of. So, you know, so almost certainly the details here are wrong. Okay, they probably weren't made out of PDAC and oligoaspartic acid. So we want to ask, how robust are the results? Do they hold up with other polyanions, for example? And Raghav was able to show that if he added a slight amount of excess polyanion to sort of release the RNA, so it didn't bind too strongly to the cation. So in other words, he's rescuing activity by adding an anion. We can see that rescue in product form, it works with backbones of carboxylates that I showed on the previous slide, as well as phosphates that are shown here, as well as heparin. And that makes sense because what's important here is the physical interactions, not the details of the chemical, which adds a kind of robustness to all of this. So along those lines, he asks, well, what if we make the coacervates not out of these sort of favorable species? PDAC was let me go back and just show for the chemists in the audience. PDAC, as we call it, is a quaternary amine. So the nitrogen has 4 different R groups next to it, which kind of makes it hard to approach. It kind of disperses the charge, but instead use arginine. And arginine is an otherwise kind of unreactive coacervate, and found that if he added the excess polyanion, that he could rescue RNA catalysis and otherwise incompatible coacervates, and that's sort of shown here with the 2-to-1 and 3-to-1, where the activity is rescued in this guy, which doesn't like to react and make it work as well as the other ones. So I want to make it clear that we don't think we understand, what I'm showing you is the details of exactly how things went down in the origins of life. We don't think it was any of these polymers per se, or even this ribozyme, but the principles of concentrating the material, of forming the protocells by associative reactions, and the robustness of these, I think, is an important point. So I want to show one last point, and it continues with part 3, getting RNA inside the protocells. And what I showed you on the first set was a functional approach, so RNA reacting. But I mentioned, and I sort of hand-waved about maybe the RNA wasn't folding very well, and we needed to add more anion in to help it to fold. So we decided we would actually go in and look at the structure of the RNA. Does it fold into a cloverleaf or not? And here we borrowed from other work that was going on in our lab. So another part of the lab has pioneered ways to look at RNA structure in vivo and genome-wide. And we developed this in plants initially with my collaborator, Sally Osman. And then we applied it not just to living cells, but to these protocells. And this is work was pioneered by a postdoc in my lab, Ryota Yamagami, who now has a lab at Ehime University in Japan, and a graduate student who now has his PhD, Macaulay or Mac Meyer, and he has a position at CSL in Boston. And what they do is use dimethyl sulfate, and I'll just focus on its reactivity with C and A. It reacts at these positions. It also reacts with G. But these are on the base pairing position. So it will react if the base is unpaired, and it won't react if the base is paired because it's blocked. So we use next-gen sequencing. There's a lot of steps. I took them all out just for simplicity on this. We've published a number of papers on the methodology. But this is a scorecard, so each row is a different one of those tRNAs that I showed you. And then we can look down and see where the unpaired regions are. And those tend to be more reactive, and they tend to be a little bit warmer, like this loop, this T loop here, some of the variable loop. And we can take these reactivities and use them to understand and restrain the folding of the tRNA. This really hot region here is because the tertiary structure of the RNA exposes this 1A and allows it to be very reactive, which gives us a hold on whether the cloverleaf structure is formed or not. I'll come back to that. So you might say, this is a pretty simple RNA. It's only 75 nucleotides. Surely you can predict this with the computer. The answer is we can't very well. So this shows the ability to predict it or in silico is only about 75%. Each dot is a different tRNA. But if we put those different chemical restraints, we can see how much the improvement is, almost to nearly 100% or 95% for most tRNAs. How can we understand that? Well, here's one of the things that the computer gets wrong. It makes this more rod-like structure. And this CCA, even though they react, they have circles, the computer doesn't know about that, and it pairs them up. When we tell it these should be unpaired because they reacted like they are here, you can see the reaction, it flips it into the cloverleaf structure. Okay, it gives you an idea of how this works. We collaborated with Dave Matthews at University of Rochester to develop a way to see how many different clusters the RNA can fold into. We did this both under normal 37-degree temperature as well as heat stress. And one of the takeaways is that under normal conditions, the RNA is mostly in the cloverleaf structure, like you would expect. There's a little bit of the rod that's predicted. But under heat stress, what happens is that the RNA, this particular tRNA, AULA, 40% of it goes into the rod form. So this was the first example where we had in vivo evidence that RNAs could misfold under stress. And I think this is quite interesting. So this person is Robert Hawley, and he was a professor of organic chemistry at Cornell University. And he won the Nobel Prize in 1968. And what Hawley showed, he was the first person to successfully isolate tRNA, and in 1964 was able to map its structure. And notice that Hawley's holding a rod form of tRNA, but behind him is the cloverleaf structure. We'll come back to that in a second. So I'm in the process of writing an article about a perspective article about Hawley's work and our work on the two forms of the tRNA that I showed you. And what Hawley said in his seminal paper in 1965 was that he provided 3 different folds for the tRNA. So he won the Nobel Prize really for getting the sequence, but he was trying to also work out the secondary structure. And you can see the cloverleaf, but you can see the rod. And he said, if you go back and read the original paper, these structures should be considered only speculative, and in any case, it's likely that the actual conformation of the RNA in solution varies with conditions. It turns out that he's absolutely correct, and that under stress, that the RNA adopts this rod-like forms. And we're writing up a manuscript right now, that tentatively has the title of Revisiting Hawley's tRNA Structural Models, where even the wrong ones were right, and his idea of them varying with conditions seems to be correct. So let me just show you a couple. I think I have just a few more slides. And I want to go back to the makeup of the coacervates. Remember I said that they're brought together by, in an associative way by oppositely charged species. And this shows the negatively charged species. They're just nucleotides, a triphosphate, adenosine diphosphate, and monophosphate. And this is a scorecard. These are 2 students from Christine Keating's lab, Fatma Kakmek and Sehan Choi. And you can see which ones form the coacervates because they have the red color. You can see the droplets. And sometimes if you push them, they make these ugly aggregates that sort of precipitate out. And so, so we took these conditions, which are, we think, potentially more prebiotic-like, because it's easier to imagine short stuff than really long things, and studied the folding of tRNA in these conditions. And this is Mac's work, who I introduced you to back a few slides ago. And we like to put things on a cube, where we have three different variables that we're changing. We can change the concentration of magnesium along this space, or along this edge, I should say. We can change the charge ratio. Remember I showed you we can facilitate folding by adding excess anion, and we can change the length. And so, to make a long story short, what Mac found was that under these conditions, which are not very favorable for folding, they have low magnesium, which RNA does not like. They don't have the excess charge ratio, that it formed the rod. But that at these other vertices, it formed the clover leaf. And it formed it to different extents. Back here at the vertex F, it's the most strongly folding, with a free energy of minus 34. And here, it's less strongly folding. We can see some reactivity along in some of the helices, which is gone here. And so if we return to Holly and the picture I showed earlier, this rod that's right here is actually quite similar to that one if you go through it in detail. And it seems that this ability of RNA to adopt these different conformations, not only in existing cells where we probed it, but also in the protocells, seems to hold, and it depends on the particular conditions. Okay, so we'll just summarize what I told you, and then look forward to engaging in a conversation with you about this. So these complex coacervates are excellent candidates for the first cells, for protocells. They form readily and with a wide variety of different polyanions and cations, including short ones. RNAs love to go into them. They concentrated about 5,000 times, and when they're so concentrated, they're more likely to do reactions. And we can enhance catalysis by adding excess polyanion to allow the RNAs to sort of peel off and not interact too strongly with the polycation. And we confirmed that by probing the structure with DMS. And that functional RNAs, like tRNAs, fold very similarly, both into native structures and rod-like structures in protocells and real cells. And I just want to give a thank you and a quick shout out to the astrobiology community at Penn State. There's an astrobiology research center that has supported us, and it's a place where students can consider coming and getting a dual degree. I have students in my lab that are both in the chemistry program, where they're getting a chemistry PhD, or a biochemistry molecular biology degree, but they're getting a dual degree in astrobiology. And then I would like to just show a picture of the group and the two people that were most involved, Mac Meyer and Ryota, who I called out earlier. And then the work that I showed you was mostly funded by NASA, but we're also funded for, I have the good fortune of being funded by the National Science Foundation and the National Institutes of Health. So I'll stop there, and I think join Professor Lazcano for questions. Yeah. Thank you.
Thank you. Thank you so much, Phil, for giving us this update on this more integrated idea of what RNA evolution could have been in the primitive Earth. You already have a remark and a question from the audience. They are watching us from in the web, in the media. And we have a question, I'll say it in Spanish, Irene NL. She says, thanks for the Friday talks about evolution. It's nourishing and illustrating. I thank the organizer, scientist, researcher. It's like a present for the world knowledge. Thanks for making it. Congrats from Southern Spain, loyal follower, which is very good. And you have a question from Paco Suarez. I suppose that he might be in Mexico or in Latin America. A couple of months ago, we saw a lecture in News from the Cosmos in the YouTube channel of El Colegio Nacional about the James Webb Telescope. Thanks to the technology, we have getting significant insights on space research. The question is, what technologies or what new observations have allowed you to obtain your, to proceed with your research?
Yeah, I mean, the space mission is not something that we directly do with our research, but we're fascinated by the Webb telescope and seeing all the exoplanets, and especially with Bennu, which was fascinating to actually fly out there, land on the asteroid, scrape up the rock and bring it back and to actually analyze it. I have to say, I was skeptical. I was worried that they were going to come back and just say they had some rock and there would be nothing in it. And when I read the article, we were writing a proposal right at the exact time that it came, and I found all the RNA that was on it. I was quite moved. Do you know what I mean?
Yeah, absolutely. I was part of the panel, of the initial panel that was supporting Dance's ideas on.
Yeah, it was emotional.
Oh, yeah, absolutely.
It was emotional actually to see that because, and so that was amazing. You know, I've been to meetings where there's discussions about what technology do we need to see signals to know that something's life or non-life, and maybe that's a little bit related to that. You know, I can only comment tangentially on that, but one of the things that I see is that whether it be on Earth through spark discharge experiments like the Miller-Urey experiment, or ones that come back and bring back to Earth from asteroids or collect them from meteorites, it's astonishing that you keep seeing, you see the 9, I think it's 9 of the natural amino acids, and you see the 4 bases. Sometimes there's not as much cytosine because of the hydrolytic deamination, but it's still seen. And it, you know, I don't want to say that life elsewhere in the universe has to be like life here, but I think there's going to be similarities in the fact that these amino acids keep popping up and these bases keep popping up, and they're stable and they have special characteristics that keep them from forming tautomers and allow them to match. So it wouldn't surprise me if that were the case. But flying to one of these exoplanets and grabbing the material and bringing it back, in our own solar system, there's efforts to do that with Enceladus and so on. So, but elsewhere in the universe, we're going to need other tools, spectroscopic tools and so on, to begin to approach that.
I'm delighted you mentioned asteroid Bennu. I was equally moved as you to use your same expression with the recent paper published about a week ago, showing that because of the salt crystals in the sample of Bennu, you can actually deduce the existence of ponds in the parent body of Bennu. I trust enormously those results because people involved, engaged in that research are Danny Glavin and Jason Dworkin, which are very dear friends of mine, and we actually have written papers together. So I was quite impressed. But something that I wanted to ask you, and I'm delighted that you raised the issue about the bases, the universality of the bases, and some amino acids. People are not paying that much attention, and it may be a case of American nationalism. I'm not paying that much attention to the results from the asteroid Ryugu that was investigated by our Japanese colleagues.
Uh-huh.
And Hikaru, this colleague of us, who's really is the head of the chemical analysis group of the Japanese group, she's very adamant about the absence of adenine in Ryugu. So apparently, you might have a diversity of chemical composition in the different asteroids.
I mean, I do know that if you look at one of the things that when I was reading the Bennu article is the balance between the purines and the pyrimidines. Mm-hmm. Of course, when you look at DNA on Earth, mm-hmm. You see A's and T's are balanced, and G's mm-hmm. and C's with Chargaff's rules. There's clear, and RNA doesn't have to follow that, but there's clear mm-hmm. violations of Chargaff's rules, both sure. on Murchison and Bennu. And I think one has an excess of purines and one has an excess of pyrimidines, mm-hmm. Which helps give confidence that this isn't just contamination from earthly nucleic acids, is one of the things that the authors point out. Why there's no adenine, I don't know. I mean, one of the things too is you see sometimes methylated pyrimidines that are showing mm-hmm. up as well, and also you can see 2,6-diamino purine,
right,
which can base pair with you as well.
Sure.
And so I didn't see the details of that particular study to see if the adenine and the extra amino group would be responsible, and maybe there was more ammonia to drive the reaction. Mm-hmm. It's actually, I mean, I think it's quite interesting. I'd like to learn more about that.
In about 1975 or 1976, Alan Schwartz, a very dear colleague who was at Nijmegen in the Netherlands, actually demonstrated that during the synthesis of, the pentamerization of hydrogen cyanide to get adenine, you could also get some peaks showing that there was 2,6-diaminopurine. Uh-huh. And in 1992, 94, Stanley Miller, Jim Clips, and myself published a paper showing that you could get 2,6-diaminopurines without any problem if you look at the solvent of hydrogen cyanide or cyanoammonia, ammonia cyanide, whatever. So that's fascinating. Well, we basically forgot about it because there was a report in the 1970s from some Russian Soviet virologists saying that you could have 2,6-diaminopurines in some fakes. And this, I think, is a very clear indication of these nationalistic prejudices. I remember that some people were dismissing this result. And I remember talking with a couple of colleagues from the NIH, in fact, saying, well, the Soviets don't have the precision and are not that careful and so on. Well, it turns out that the first year during the pandemic, a very large group of very, very capable virologists, chemists from France, headed by Philippe Marlier, your namesake, in fact, demonstrated that you have fakes that have 2,6-diaminopurines, which is amazing. Yeah. Apparently, it might have evolved as a way of resisting the E. coli immunological systems and so on. Yeah. But there you have a very clear indication that in functioning biological entities, you can have 2,6-diaminopurines. I'm very keen on that molecule myself. So for reasons that, Ricardo Hernandez Morales, who's here, can explain you why we are so keen on adenine ourselves and on 2,6 and its derivative. But it's fascinating that, as you point out, you can have different reactions that can lead to it.
I mean, what happens, of course, as you know, when you have the 2,6-diaminopurine, is you sort of level the playing field between the two base pairs.
Right.
Because normally you have a GC, which has 3 hydrogen bonds, and the AU, or AT, which has 2. But when you bring 2,6-diaminopurine in, now you can have 3. Right. And so the energetics are much more equivalent, which changes the situation.
The stability of the molecule.
Yeah.
Right.
Yeah.
So I'm delighted that we are tuned about with the same molecules. But of course, we have chances to have questions. Por supuesto que tenemos espacio, tiempo para preguntas o comentarios del público que está aquí con nosotros y desde luego quienes nos siguen en las redes.
Hola, I'm going to try to speak English. I'm not very well speaking English, but thanks for the talk. And tRNAs are in the cell. tRNAs are part of coacervates. Yes.
So we don't know if tRNAs are part of coacervates. What we see in modern cells is RNAs are often part of coacervates, and they're often unstructured RNAs. And I don't know that tRNAs are part, but it's possible that there's some data that I'm not familiar with. People like Roy Parker at Colorado has looked at the RNA compositions of P-bodies and so on to see what's present in there. They tend to be, I didn't say too much about this, but you can imagine RNA having two roles in coacervates. One is they can make up the coacervate and the associative interaction between the cation and the anion. There we imagine that those RNAs would be pretty unstructured. And then we have the ones that go inside, that we're doing chemistry, that we were able to rescue. So you almost could think of them as being a structural RNA, maybe an unstructured RNA and then a structured RNA, and the unstructured kind of forming the droplet, and then the structured ones going inside of it. They're all inside of it, but going inside and doing chemistry and so on. So we can think about these things from the point of view of prebiotic systems, but we can also think about them and take clues from biotic systems as well.
Okay, thanks. Because I used to think ribosome and the translation machinery as a coacervate, because I don't know, it's made of RNAs and proteins, and they get together to make the translation process, right? So I like to think that ribosome is kind of a part of a coacervate. So tRNAs are like part of that system, right?
Yeah, I think, we were recently doing some structure mapping with new reagents on RNAs, and we found that they were forming droplets. Because up until 10 years ago, no one thought about this. At least I didn't, most people didn't. There were probably a few, O'Parron was thinking about it 100 years ago, but most people were not thinking about it. And we just flicked the Eppendorf tube and look at it, and it was cloudy. And so we could put it under the microscope, and we could see that it had phase separated into droplets. And so your idea that the ribosome and other systems could form coacervates of sorts is absolutely possible, and I think we should be looking more to see if they're doing that. I mean, one example that I find interesting, and I learned about this a number of years ago, I want to say 20 years ago, something as simple as methanol in water does not fully mix. And that seems shocking. We know it's miscible in all proportions, right? But if you do neutron scattering on a water methanol mixture, the methanol forms little icebergs, little puddles, and like 4 methanols, the methyl groups all come together, and the OHs interact with water. So something as simple as that is not mixing. We have to wonder how much of this is going on in the cell everywhere. Yeah. Or protocells.
What I like fascinating about this exchange is that until a few years ago, even today, we teach and we believe that the inside of a cell is just like a bag full of water and some metabolites and some molecules, but clearly, there is some internal structure due to physical-chemical interactions that really make the inside of a cell something much more complicated than just a water solution or a cytoplasmic solution.
Right.
Yeah, I think that's fascinating. Dr. Campillo, if you please.
Thank you for your...
I think it's the first time that Dr. Campillo is asking a question here.
Thank you for your talk. I think number 4, there are some problems still, right? I mean, about the dynamics of the RNA folding. I don't know if because to have an approach in vitro or in vivo are very different because of the physiological conditions, right? What about, I don't know if there are studies about, for example, the in viroids, that these biological entities that have RNA in their genomes, but some of them behave like ribozymes, for example. So it's RNA, infectious RNA, and I don't know if there are studies about how it is the folding in these biological entities. That's one question. And the other, if there are deep learning approaches to predict these RNA structures, secondary RNA structures. Thank you.
Yeah, excellent questions. My lab is kind of trying to approach it from 2 directions, top down and bottom up. And top down, this is the work that we've been working with our collaborator, Sally Osman, in biology at Penn State, Paul Babitsky in biochemistry and molecular biology at Penn State, where we actually take cells, whether they be from a Arabidopsis or a crop like rice, or bacteria, gram-positive and negative, and even in archaea, and bring chemicals in and probe how the RNAs fold. It also can be done in viruses. My lab hasn't done very much, but there was a flurry of stuff that came out right during and after the pandemic on the folding of SARS-CoV-2 and how it folds. And there's a lot of things that we don't understand yet fully, cryo-EM and so on, all of those are helping. So that's kind of top down. And bottom up, in that case, we start with water and add stuff to it and try to build up the components of a cell to see how they begin to affect RNA folding and structure, and maybe we can meet in the middle. Our lab has been working on some kind of mixture that we call ECO80, and ECO for E. coli, and it has 80% by mass of the metabolites. And so we add all these things in, it's kind of a complex undertaking, and we're able to capture a lot of the interactions of magnesium with metabolites and so on, and we see how RNA folds there. But I think, as Tonio said, it's and we've discussed, the cell is really quite complex, and capturing that is not trivial. And so, each approach has its own advantages. Using the cell seems like the only way to go because it has everything in it. But the problem with working in the cell is like chemists like to change the conditions, and we want to turn the knob and see what happens in response. And it's hard to do that in a living cell. It's hard to change the pH or the ionic. So the bottom up allows us to do that. So it's really the combination of the two. And I'm confident that what we know is just scratching the surface of what's left to be understood.
I do have a question myself regarding the idea of protocells. Quacerbates, I was quite close to late Professor Opperin, personally, scientifically, and of course I understand the significance of quacerbates. And you have the tremendous advantages of quacerbates that you can go inside or outside, inwards or outwards of the quacerbated cell without any major problem. On the other hand, I also like my lipids.
Right.
Because if you have a membrane bound by lipids, or the possibility of having liposomes in the primitive Earth, I'm thinking of Dave Dimmer and Pierluigi Luisi and Jack Szostak, Irene Sheen, and so on. You have something that's more similar to what we see in cells.
Yes.
But it's not so easy for nucleotides or amino acids or a number of things to go through the lipidic membrane.
Right.
How will you reconcile the idea of quaterbates with not only the properties, but the concept of quaterbates with the idea of liposomes or micelles in the primitive Earth?
Yeah. We're thinking about that with some of my colleagues, and it's there's a number of different scenarios that one can imagine where lipids form and they encapsulate inside of them the species that are needed for associative coacerbation, so that coacerbates can form inside of them. And another question is, how do you get the nucleotides? How do you get the RNA to go inside? And especially once they're formed, it's really hard to get them in. And I mentioned that. They're not very permeable. They have often the same charge, negative charge, so they're repelling each other. And so the idea that somehow when the conditions are right, they can sort of engulf all of that, whether it be at the surface or it be at certain conditions inside of the early Earth, specialized environments that one can try to imagine where it sort of forms and captures it on the inside. I think we need to start to look into that and what conditions might have made that possible.
But this idea of having a liposome encapsulating a quaterbate is very attractive. It's very eclectic, but it's also very attractive, right?
Yeah. I mean, I think it's attractive. I think, and that it captures a lot of the features of current cells, which of course have lipid membranes, and then they have organelles inside, which also have lipid membranes, but then they also have coacerbates or liquid-liquid phase separations inside of them, as we know, P bodies and things like that. So it is a way to sort of do that until we have a system that can synthesize those internally or selectively allow import and export of those. But until something so sophisticated has evolved, one maybe can imagine some sort of way in which it's able to capture that inside of it, so that you can get both of the things that you love and that we love inside of a single system.
Right.
Because I listed each had disadvantages.
Sure.
But they were. The disadvantage in one was an advantage in the other, and so in some ways, if you could have them both, you would eliminate most of the disadvantages. So your point is really well taken.
Just for the historical anecdote that you might find interesting is, in 1948, Operin was actually working with RNA, with quaterbates with RNA. And you can find a reasonable report in that old series that NASA used to have in the 1970s, 1980s. They used to translate papers from Russian into English. And they're absolutely fascinating. And once I was discussing this thing of the RNA inside quaterbates with Michel Morange, which is a very, very distinguished historian of molecular biology. And he pointed out something that was quite interesting. Operin at the time couldn't accept the idea of RNA being a genetic material. But you have a polyanion, and for him working with quaterbates, there were polyanions and polycations. It was the perfect choice because it was very easy to work with, to have. So on the historical side, I think that's quite interesting.
I agree, completely agree, yeah.
And then he actually tried to prove that if you add ribonuclease inside the quaterbate with RNA, the reaction would be much more efficient than, of course, in the outside medium, right? So that's fascinating also. You have a question, another question. Oh, by the way, Chloe, you have some congratulations from some of your students for having such a bright father explains things so easily. So you also get to be noted in the lecture. And we have a question from Adrián Quintero Martinez from the Institute of Chemistry at UNAM. And his question is, what could be the implications of knowing that these RNAs can misfold under stressful conditions? Could we use them for controlling any pathogenic species?
Mm-hmm. Yeah, it's a great question. That's really looking more into life as we know it now. One could imagine, for example, we speculated in the work that I showed, it was published in PNAS in 2022, the Yamagami paper where the tRNAs began to misfold at the high temperature, that might be a way for the E. coli to cope with the temperature stress. And so, but not every tRNA went into the rod. Some did not. And that there may have been a program there. And interestingly, the two of the ones that did not were FMET tRNAs, which initiate translation as if maybe there was a need to get translation going. And then some of the common ones were misfolded and then halt it. Okay. And then maybe allow a way for to reactivate translation once the stress had passed. But beyond that, roles for roles for RNA misfolding and disease in general, it's a really good question. There's certainly a lot of therapy that is being done with RNA and RNA folding through CRISPR gene editing. Oh, yeah. Nobel Prize, I think, 5 years ago. RNA is really a future in medicine. Certainly if the Nobel Prize 2 years ago in the mRNA vaccines and the modifications to suppress the innate immune response, and then just this year in microRNAs, which can be used to turn genes on and off. You know, most diseases that are treated by drugs, they're small molecules that bind to proteins. And so there are a number of people, Matt Disney in particular, and others that have led the idea of drugging RNA with small molecules. But if we can target RNA in general, we can target so many diseases, including undruggable ones at the protein level, also with base pairing to the RNAs. And so so it's a really excellent question. I think there's many things that we'll probably learn that we don't currently understand. But I showed the beautiful pictures of RNA folding, and when RNA can't do that, that can lead to disease. So it's important to understand, just like protein misfolding can lead to disease. We don't know enough yet about that with RNA.
Regarding that, can I ask a silly question or naive question? I should know the answer, but I have never worked with extremophiles. What about heat-loving prokaryotes? How do they prevent the misfolding of the transfer RNAs?
Yeah, we actually studied that a little bit some time ago. Published a paper looking at the base pairing content and stability of tRNAs across the different temperature ranges that they live. And what you find is that the ones that are really heat-loving, that those ones tend to have very GC-rich helices, which right away you say, oh yeah, that makes sense. Sure. But then when you think deeper about it, it actually doesn't make sense too much, because they have to have tertiary structure. And once you lose the tertiary structure, if you open that to a GC-rich cloverleaf, it doesn't really help the situation very much. And so it turned out to be that the RNAs seem to fold cooperatively. So that they don't populate the intermediates. It's more all or none from a tertiary fold, which to an unfolded state. So they're two state-like, and that tertiary fold has GC base, has the base pairing in it. And so it seems like one of the simplest ways is through strengthening base pairing if the folding is cooperative.
Oh, okay.
Yeah.
Which again, we normally think of a molecule in isolation and not as part of a complex system with additional components.
Right.
That can lead to cooperative folding, for instance, right?
Yeah, and one of the things that we're learning too, and we still need to do more experiments on this, the bottom-up approach where we're building up the cytoplasm with the ECO80, is that those metabolites tend to slightly weaken secondary structure, and that actually would enhance this cooperative folding by destabilizing those intermediates.
Could molecules like polyamines play any role in stabilizing 3D structure of RNAs, transfer RNAs under stressful conditions?
Yeah, it's a really good question. I mean, molecules like spermine and spermidine certainly are known to stabilize RNA tertiary structure. These are molecules that could be +3 or +4, but they're not concentrated into like aluminum into a single nucleus. They're spread out, and so they can spread out and stabilize the RNA. I don't know. I think we need to learn more about that. But if we look at crystal structures that have been solved of tRNAs and other RNAs, we'll often see them coordinated with these polyamines.
Well, now we have before going to this question, which is a little bit more ideological or political from the audience, I'm sure you will not mind. I would just like to recall the excellent work of Dora Tang, an extraordinary researcher. I think she's
I know Dora, yeah, she's
from the UK, and now she went to Dresden, I think, and she's doing beautiful, beautiful work on conservation.
That's right.
Yeah. And so having said that, we have a question from Mr. Carlos Garcia. How compatible these findings back on research are with the belief that life was magically created, which is a view mostly shared by the group in the highest of current USA government?
Yeah, I mean, there is a certain level of controversy one could say about, did life come from matter and simple molecules or through some divine intervention, perhaps, right?
Right.
And so
it's not that question, it's from Mr. Carlos Garcia.
I mean, some people who think deeply about this have come to peace with this. I believe there was a papal conference on this, in which the Pope said that, something like truth cannot deny truth, and that if God put into motion the universe and the molecules that were needed to allow this to happen, to move from matter to life, then so be it. Then that's the way,
But the Pope is quite different from Mr. Trump.
This is true. This is true. But the idea that science and religion can't coexist for some people is not a problem. For others, it might be a problem.
Right. Well, I'm sure we all agree that only in a democratic system can these wider spectrums of beliefs can coexist. And so, well, thank you so much, Phil. Thank you so much, Professor Bevilacqua. And thanks, Chloe. You see, you get also congratulations. There are a number of people that have called or written congratulating you for the lecture, and we normally read their names. So, well, we have to thank the School of la Escuela, la Facultad de Enfermería y la Coordinación General de la Extensión de la Universidad de Colima, al Instituto de Fisiología Celular y al Laboratorio de Origen de la Vida, porque nos ayudaron en la transmisión de esta presentación. And you have greetings from Ricardo Mariño Pérez from Veracruz, from the town of Juan Rodríguez Clara in Veracruz, Laura Meneses from Mérida, Yucatán, Ángela Peña from Guadalajara, Olga Sánchez González from Guanajuato, Federico Certuche from Monterrey, that's a whole spectrum of the country, from Mexico City, Juana Herrera, Marcela Elizalde, Noemí Edith Marcial Flores, Iracema Portos, Luis Fuello McDonald, un buen amigo, Marcela Sánchez Delgado, de la Facultad de Medicina del Estado de México, de la página web del Seminario de Unidad del Desarrollo Académico de la Facultad de Medicina de la UNAM, de Texca Tatevari, del doctorado de la Escuela Normal de Estudios Superiores, la Escuela Nacional de Estudios Superiores de Morelia de la UNAM, de Ale Talango, del Instituto Campechano, de Alberto Pacheco de la UNAM, de Nidia Caamal, de la Maestría de Ciencias y Tecnología Farmacéutica en la Facultad de Química de la Universidad Autónoma del Estado de México, Carolina Farías, is sending her greetings from Colón in Argentina, and she says that your lecture was the best birthday gift that she could get today. Antes de terminar, les quiero invitar a una mesa redonda que vamos a tener aquí en el Colegio Nacional, una mesa redonda presencial el martes 18 de febrero a las 12 horas. Es una charla que va a dar la doctora Carmen Rodríguez Armenta de la Secretaría de Educación Pública sobre un problema central y es educación e inteligencia artificial, retos y oportunidades. La mesa redonda va a estar moderada por nuestra presidenta en turno, la doctora Susana López Charretón, y participan el doctor José Ramón Cosío, el doctor Luis Hernando Lara y sus servilletas. Y dicho eso, no me queda más que agradecerles a ustedes su presencia, and thank you so much, Phil, for accepting the invitation for being here. It's quite an honor. Thank you so much.
Thank you.
So, goodbye.